but we could provide it exogenously, that is, not a normal compound, but let's say we inject it, we get extra epinephrine. Whatever your state of epinephrine in your body, you're gonna have more of it, greater response, okay, heart rate goes up. Relaxing on the beach on a hot day. Relaxing should lower your heart rate, not elevate it, right? And we'll talk about how that happens in a moment. And then you're on a hot day, so that is additionally going to have the effect of lowering thyroid hormone levels. Yes? The biggest sort of important thing that it does is that it will cause the relaxation of your airways. So particularly people who are having an allergic reaction, one of the major concerns is that your airways are going to constrict. And that's a result of histamine and a few other chemicals that are hyper-responsive. Okay? Epinephrine causes the dilation of airways. Okay? That's why people very often will use an EpiPen, or one of the main reasons. Okay? We don't necessarily want your heart rate to go up. It's a side effect. But as part of the fight or flight response, your airways dilate, they get bigger to allow you to exchange air more quickly. So we're taking advantage of that mechanism. Okay, so the M2 receptor, this is part of the parasympathetic nervous system's control of the heart rate. Okay, so this one's actually very simple, except for a couple of things where it's not. Okay, so acetylcholine comes along, and it binds to the M2 receptor. Okay, and judging from the way I've drawn it, what does it look like? What kind of receptor? It's a G protein coupled receptor. Okay, it passes back and forth through the membrane, right? And it's coupled to a G protein. One thing I want to call your attention to is that we have here two different types of G proteins, and one has an alpha S subunit, and the other one has an alpha I subunit. I stands for inhibition, and it will inhibit adenylyl cyclase. S stands for stimulation or stimulatory, and it stimulates adenylyl cyclase. Okay. So when we talk about these receptors, very often we'll call this G protein coupled receptor, we'll call this a GS receptor because it has the alpha S subunit. And this one over here we'll call a GI protein because it's inhibitory. It has the alpha I subunit. Okay. The beta gamma seem to be similar, but what makes these unique is whether they're bound to an alpha I or an alpha S. Okay. So it's a different second messenger system, but it sort of intersects, right? And basically does the opposite. All right. So let's take a quick look at this. All right. The alpha I subunit takes on a GTP, becomes active, and it binds to and then inhibits adenylyl cyclase. Okay, so whatever cyclic AMP might have been being generated is now stopped, right? So we can't make any more cyclic AMP. Okay. Another thing that it does, not exactly sure how, is that it activates two important enzymes. And these you may or may not know about, depending on how much cell biology you have, but a phosphatase is sort of the mortal enemy of a kinase. Okay? So if a kinase is responsible for phosphorylating things, a phosphatase takes phosphates away from things, from proteins. Okay? And these are always working in the cell. Okay? So there's always a little bit of kinase activity happening. It's not black and white. There's always some happening, right? And then it's being reversed, right? So the phosphatase comes along and sort of undoes what has happened due to the kinase. Does that make sense? You've got a roommate like this, right? Pick something up, clean up after yourself, and they come along and make a mess of it. Or at least that was my experience. Larry, his name is. He had a crush on this girl who I initially married. We won't discuss it. All right, phosphatase. So that's a phosphatase. It is the opposite of kinase. And then there's phosphodiesterase, which comes along and breaks apart cyclic AMP. Okay, so it's cyclic. All right, and the phosphodiesterase comes along and it turns it back into a linear structure. Right? So it uncyclifies it. Right, so it just becomes AMP. Okay, so these enzymes, I don't want to give you the impression that they're there, they weren't there before. They were there, they were working at a low level, sort of undoing the activity of kinases, right? And then all of a sudden, with the M2 receptor stimulation, we elevate their activity, okay? So this probably happens from the beta gamma subunit, but don't worry about exactly how it hasn't even worked out entirely. Okay. So we're elevating this activity. Why is this important? Well, it basically undoes the production of cyclic AMP, right? And it undoes the kinase activity. Okay, so it's not enough just to stop cyclic AMP production, right? That's not going to slow your heart rate. Because these things are already phosphorylated. So it undoes that. And probably the most important part of this is this beta gamma subunit comes over and it binds to our potassium channels. Okay? We've got some potassium channels here that we control, and we increase permeability of potassium channels. Okay, and these potassium channels work during the pacemaker potential. Okay, so instead of allowing more positive ions in, like norepinephrine does, now we're going to be allowing more positive ions out. Okay, different ions, but we're gonna have potassium leaving the cell instead. Yeah.
So would that mean for the one that you showed us that it would have a longer pacemaker potential?
Yeah, let me back up to that picture. I didn't put one in, unfortunately. So if we have potassium instead, there we go, potassium channel that's now open. What's going to happen is that the pacemaker potential is going to take longer. It's probably not going to bend like that. It's going to take longer to get there. So let's do it in a straight line. So maybe it takes that long to get there, right? And then we have and then another slow rise. So the potassium that's allowed to move through the cell membrane is offsetting what's happening with HCN. So with HCN, there's sodium coming in, right? And we have calcium coming in, bringing positive ions to the inside. That's moving us up. And what this potassium does is it offsets that, right? So potassium is leaving, and so the gain of positive ions on the inside is reduced. Okay. So we're still moving towards threshold, but we're doing it more slowly, right? So that's what this potassium channel does as the major target of our M2 receptor. Okay? So parasympathetic nervous system is going to slow us down through elongating that pacemaker potential. And the sympathetic nervous system is going to speed us up by shortening that pacemaker potential. Okay. All right, so one other thing, don't write this down, it'll blow your minds, okay? So we got all this antagonism happening, right? We've got antagonism at the level of adenylate cyclase. We've got antagonism breaking apart cyclic AMP, generating it. We've got kinases and phosphatases working against each other, right? These channels with their positive ion flux in opposite directions. So there's a whole bunch of antagonism that's happening inside the cell between what was initially started by the autonomic nervous system, right? And you remember the sympathetic and the parasympathetic nervous systems have these antagonistic actions very often, okay? So it extends beyond that, so not just inside of the cell. Most autonomic nervous system targets will actually have little collateral branches of their axons come over. So from the sympathetic nervous system, it'll release neurotransmitters that inhibit the activity of the parasympathetic nervous system. Okay? And the parasympathetic does the same thing, right? And you can even take this up into the brain. The centers that control sympathetic and parasympathetic at the level of the brain very often antagonize each other. Okay, so it very much enforces you either have one type of signaling or another, right? And they sort of battle it out, and whoever wins wins. Okay, whoever has the greater strength wins, but whoever wins also effectively shuts down the opposite effect. Okay, so complicated, right? What type of receptors do you suppose would be on those neurons and their synaptic terminals? The other G protein coupled receptors. Okay. So that's how we control heart rate. Simple, right? That was a joke. Come on, people. All right. Stroke volume. There are three things that affect stroke volume. One of these happens because of the nervous system, and that's contractility in the middle there. Okay? And we define that as the degree of contraction, the amount of tension that's generated. Okay? And then there's these two other things that are happening automatically. We don't control them. They're just properties of the way that the heart works. Preload has to do with the amount of stretch that happens to the ventricles. So do they contract when they're really stretched or not so stretched or not at all stretched? That impacts how they function. And then afterload is dealing with how the ventricles contract when we are opposed by a pressure in the aorta. Okay? So when we contract against a high pressure, we move less blood than if we contract against a low pressure. Right? All right, so we'll talk about these concepts. We'll start with preload. Okay. So to explain preload, the idea that we have the degree of stretch controlling how much stroke volume we produce. To explain this, I want to turn again to skeletal muscle. Okay, so in a skeletal muscle, you have this relationship that we often refer to as the length tension relationship. What this basically means is that when a muscle cell contracts, the amount of tension it produces is strongly correlated to the initial length of that muscle cell. Okay. So if we have a muscle cell that is sort of squished, very short, it's going to produce less tension than if it's stretched out a little bit further. Okay. And if you stretch it out too far, then the tension goes down again. Okay. So this assumes the very same nervous input to that skeletal muscle. Right? So we're not stimulating it more. So not all contractions are going to be the same strength. Just one of the things that affects muscle cell contraction is their starting length. Okay? And this has to do with sarcomeres. Okay, so what's a sarcomere? You guys remember? How do you define sarcomere? Your hearts have them, skeletal muscles have them. Yeah.
Basically just a muscle unit.
It's a muscle unit, a unit of contraction within the cell that is made up of actin on one side and actin on the other side, and in between them myosin. Okay? So we got a very simplified version of that shown here at the bottom, but this might represent one sarcomere. Okay? The same sarcomere might have lots and lots of actin and lots of myosin, but they have a certain longitudinal arrangement like this. Okay? So in the middle, you can see in red we have myosin. These little myosin heads grab onto actin, right, and pull. Remember this? And when they pull, they have the effect of shortening that sarcomere and generating tension. Okay, that's the general idea. So in the middle at C, we have the greatest amount of tension. And the explanation for this is that we have an optimal alignment of those myosin heads to actin. Okay? All the myosin heads can grab a hold of actin and pull. Okay, sounds great. What happens if we stretch that muscle out, however, and then start contraction, is that you can see in D, we've lost some of our myosin heads, right? So there's fewer myosin heads able to grab a hold of actin. Okay? And that makes the tension generated weaker. Okay? And you stretch it out absurdly to the point where those myosin heads no longer contact any actin, the tension drops significantly, right? So it makes sense that the more alignment you have, the better. The other direction, what happens is that basically these actin filaments that are separate from one another come together and they interfere with one another a little bit. So that maybe the myosin is grabbing the wrong actin, right? The actin that's supposed to be from the other side, right? So there's like an interference that takes place. Does that make sense? You see these little blue lines kind of interfering with one another. So whether we have the muscle too short or too long, we get a low amount of contraction. And in the middle, we have optimal alignment. Okay? And again, this is the same action potential producing different amounts of tension depending on the length. Okay? So our muscles work this, your bicep muscle contracts most strongly in the middle of its range, right? So that 90-degree angle, that bicep muscle is stronger, it generates more tension than when it's fully extended or fully flexed. Okay? So the heart follows kind of this same idea. Okay? But what we see in the heart is that it has sort of a cutoff to it. Okay? It has a cutoff to the curve. It's the same curve, more or less, but it's cut off on the end. And the reason that it's cut off on the end is that there are a couple things that restrict the heart from stretching too far. One of those is external to the heart. Anybody remember the name of the little sac surrounding the heart? Pericardial sac. There's a fibrous layer of the pericardium which limits how far those ventricles can expand. Right? So if you're just dumping all kinds of blood into the ventricles, they can only expand so far. Okay? So that keeps individual cells from not getting stretched out too far. And the other thing is that within the heart, there's a lot of connective tissue. Okay? And that connective tissue is tied into these cells, and the connective tissues sort of limit the ability of that heart to expand. Okay? So you can think of it as a heart cell is kind of like a rubber band. Okay? You can stretch it easily, and then all of a sudden you reach this point where you can pull, but it stops stretching, right? And that has to do with the physical makeup of the connective tissues of the heart, okay, that limit that. So we have the same relationship here, but in practice or in normal, physiological situations, we just don't go past into that next decline of the contraction strength. Okay. In fact, mostly when we think about this idea of preload, we're dealing with things in this sort of range of this tension curve. Okay. And so what this means is that there's almost a linear, but certainly a proportional relationship here, where the greater the length of the cell, the greater the amount of strength it has. Okay. So if we take an individual cardiac muscle cell and stretch it out, it's going to be generating more tension than one that is short. Okay? And so that's what's happening at the cellular level. Okay? But we know the cell is more than just a bunch of little cells, right? They're all interconnected into these long chains that wrap around and form the ventricle. Okay? And so that cellular phenomena, as it translates to the ventricle, at the level of the ventricle, we can see that, well, what's going to stretch those cells? What's going to make those cells long? Well, it's going to be the volume of the ventricle, right? So if we have a greater volume in our ventricle, those cells are going to be stretched out. Okay? If we have a small volume in the ventricle, you're going to have cells that are bunched up. Okay? And I'm talking here about the time just prior to contraction. Okay, so if we want to be specific, we might use the term end diastolic volume to be accurate. So the more the heart fills up with blood, the bigger the chamber, right? And so the bigger the chamber, how do we get a bigger chamber? Well, the cells are stretched out. Okay, so as we fill the chamber with blood, the cells get stretched out. Okay? So when we think about what's happening here is that the volume that's ejected is going to be increase with increases of end diastolic volume. Okay. This is known as the Frank-Starling law. That's two people, not one. Mr. Frank, Mr. Starling, Dr. Frank, Dr. Starling. And we could say that the stroke volume, the amount that's ejected, okay, related to contraction strength, is very strongly proportional to the end diastolic. Okay. So, push a bunch of blood into the heart, what happens is the heart contracts more strongly. Okay, that's the concept of preload. Any questions? Is that straightforward enough? Okay, so we have a length tension relationship that's similar to sort of the first half of what we see in a skeletal muscle cell. Oh, that's weird. Okay. Any questions about preload? Let's jump to contractility. I gave you a break from cell biology, so now we can come back to it. Very similar thing. Okay, we're dealing with contractile cells now. Norepinephrine, our GS protein, right? Cyclic AMPs produced, protein kinase A activated. And now we have targets that are a little bit different. So each cell can have different targets to PKA. Okay. So one thing we do is we phosphorylate myosin. And phosphorylated myosin knows to contract more strongly. Okay, so we phosphorylate it, and the result of that is this molecule works more effectively. Okay, so with norepinephrine around, those ventricles will contract more strongly because myosin is more active now. We have an increase in intracellular calcium, okay, because we have phosphorylated our L-type channel. What part of our action potential is the L-type channel responsible for? Plateau. Okay, so more calcium comes in, right? Binds to the ryanodine receptor, right? Releases more calcium to the troponin. Okay, so we get a stronger contraction for that reason as well. Okay, any questions about these two things? It's about to get weird. It's not that weird. The last thing is there's a protein, a new protein you've never heard of. Maybe you have. It's called phospholambin, and it gets phosphorylated. And phospholambin is bound to this pump. Okay, and what is this pump? It is a calcium pump in the SR. So what do we call it? Remember? Yeah, that's right, SERCA. The sarcoendoplasmic. Remember that? Sarcoendoplasmic reticulum calcium ATPase. Okay. So what's its job? Okay, this does not function during contraction. Okay, what does it do? Who remembers? Yeah.
It moves calcium back into DSR after it's flooded due to contraction.
Okay. So we're going to have calcium coming into the cytosol from outside, and we're going to have calcium going to the cytosol from the SR. If we supercharge our calcium ATPase, what happens is that we get much, much more of that total calcium in the cytosol packed into the SR. Okay? So the effects of this, at first you might say, well, the effect is it's going to make relaxation happen more quickly, I guess, and maybe it does. But what it does is it elevates calcium in the SR. So what happens next time you open up the ryanodine receptors? You're gonna get even more calcium coming out, right? Because you're gonna have a strong gradient pushing calcium out, okay? So, 3 things happen as a result of norepinephrine. Activate myosin, activate calcium channels, and then we activate or increase, we can say, calcium storage in the SR. Okay. So with each release of calcium, we're releasing substantially more calcium to the troponin and getting more contraction. Okay. Crazy stuff. All right, so what does the parasympathetic nervous system do to the contractile cells in the ventricles? Nothing. Remember? No parasympathetic innervation of the ventricles. Okay? So we can just move on, right? Agreed. Let's do it. Okay, so with any degree of sympathetic stimulation, we're going to get increased contractility. That's what this chart is showing you. So right here, okay, got a normal resting value of a normal amount of stroke volume, right, or we could say tension produced for contraction. If we have sympathetic stimulation, that goes up, right, because we've activated myosin, we've increased intracellular calcium. Okay, if we move over to here instead, right, let's say we've got a greater preload. Okay, we've got a greater preload, right, and we have sympathetic stimulation. So preload alone brings us to here instead of here. And then the sympathetic nervous system works on top of this. Okay, so both preload and norepinephrine work on the idea of increasing the stroke volume. Okay. Very quiet. Any questions? Yeah. Could you say that just a little louder, please? And yeah. Okay.
What regulates the amount of phosphate that's already taken here so that myosin and active interaction occur?
What determines the amount of phosphate? So you're referring to let's see if I'm on the same page. This here we're talking about the phosphorylation of the myosin. And you're asking what determines how much phosphorylation there is?
Or what allows the phosphate, what to balance,
Okay, all right. So there's protein kinase A, which is going to have a low level of activity, okay, if it's unstimulated. And there's also going to be a low level activity of phosphatases, okay? So in every cell, make sure I can spell this right. Okay, so phosphatases are antagonizing this phosphorylation process, or maybe it's better to say they're removing phosphates. So phosphates are moving away, and kinases are adding phosphates. Okay, so if we completely remove all norepinephrine signaling, you will have a low level of both of these. You'll have a low level of pKa, phosphorylating, you'll have a low level of phosphatases, dephosphorylating. And so they'll reach some balance, right? Some homeostatic or antagonistic balance, okay? When norepinephrine comes along, what happens is we temporarily tip the scales, right? We're elevating pKa activity. So for a time, there's pKa activity is going to overwhelm phosphatase activity, and you get more phosphorylation, okay? And when that stimulation goes away, pKa activity drops back to where it was, And over time, not immediately, but over time, you'll restore that balance to where it was. The phosphatase activity will sort of eat away at those phosphates and it'll come back down. Yeah. So, and I'm glad you brought that up. A lot of times this goes unsaid that, well, how do we undo it, right? And there are these sort of processes that are happening at a constant level. So just like contraction is the release of calcium into the cytosol, well, there's a mechanism to take it back out. And it's always working. It's just, we have these sort of antagonistic properties of the cell machinery. Great question. Okay, so let's talk about afterload. This is one that is a little bit complicated, but not I don't think it's so bad. If you can understand the contractility thing, you're home free. Afterload. Okay, so this is the force opposing ventricular ejection. Okay, so unlike the other two, this is inversely related. Okay, so if you have high contractility, you get high stroke volume. If you get high preload, you get high stroke volume. Okay, or low. Okay, afterload is has an inverse relationship. So the greater the afterload, the lower the stroke volume. Okay. So how this works has to do with the concept of velocity. This is a little bit weird, but again, let me draw an analogy to help you understand this. If I have a dumbbell that weighs 1 pound, okay, and I want to shorten my bicep muscle as quickly as possible, okay, I'm going to be able to do that really quick, right? There's a high velocity of that bicep muscle shortening. If I go up to 5 pounds, 10 pounds, 25 pounds, my ability to shorten that muscle gets, I still have the ability to shorten, but the time that it takes is longer, right? The velocity decreases. And at some point, probably not a very high amount of weight, I won't be able to lift it. Okay, suddenly 50 pounds or whatever, maybe I can't lift it. Okay, so my velocity of shortening goes down to 0. Okay? Same idea here. Afterload, as we define it in skeletal muscles, is really the resistance that's placed on the muscle. Okay, the amount of force opposing contraction. Okay, and we can apply that to the heart too. So when we have a heart that has low resistance to it contracting, that ventricle can spend a great proportion of its time moving volume out. Okay, but if we raise the aortic pressure really high, now what's going to happen? Well, we go through that isovolumetric phase. Remember this? And what are we doing in the isovolumetric phase? Raising pressure, right? We now have to go to a greater pressure than we did before. So the amount of work that we're putting in for that one contraction, a lot of the effort is spent raising pressure, and less of it's spent moving volume. Okay? So let me illustrate it like this. Okay? Here's our old friend, the pressure volume loop. You love it. You know you do. All right, so those two parts on the side, the isovolumetric contraction and the ejection. These represent ventricular systole, right? Okay. And one contraction, one beat of your heart is the result of an action potential, right? So it's not summation and you need to put in more effort or anything like that. Okay. It's one action potential and we get one contraction. Okay. And it's a matter of where does that effort go? Does it go to producing a higher pressure and less volume movement? Or is it less pressure and more volume movement? Okay. In terms of the physiology of this, in physical terms, we call the total amount of work that's done the stroke work. So the amount of work that that ventricle does with one beat of the heart, that's the stroke work. Okay. And there are two parts. We have volume work, where we're pushing blood out. Right, so the tension that's produced goes to moving blood out. And then there's tension that's produced, work that's done, that is for the purposes of raising pressure up to the aortic pressure. Okay? So in other words, you've got a defined unit of work that's being done, the total stroke work. If you take away volume work, you've got to have more pressure work. Okay? If you take away pressure work, you're gonna have more volume work. Make sense? Okay, so when it comes to our blood pressure, with a high blood pressure, in other words, a high afterload in the aorta, one contraction of the ventricle accomplishes more what? If we have a high pressure, what kind of work do we disproportionately produce? This is pressure work, right? Because the aortic pressure that we're pushing against requires more of the work that's produced into producing pressure, okay? And less volume work, okay? So, I've got one other thing I want to talk to you about on this topic that we'll talk about next time, and then we'll jump into blood vessels.